higher to low. In active transport, we're going to transfer material from a lower concentration to a region of higher concentration. So we require energy in the form of ATP. This energy will be utilized to induce a conformational change in a carrier protein to transfer this material across the plasma membrane. When we talk about active transport, it's to be carrier mediated. And we say carrier mediated. What do we mean when we say carrier mediated? Carlos. When we say carrier, Deidre. All right. When we say carrier mediated, active transport is carrier mediated. Vivian. Transport membrane. Right. So we say it's carrier mediated. It requires a transport protein. So active transport is carrier mediated. It requires a transport protein. There's two types of transport. We have primary and secondary transport. And we'll see the difference between the two based on the requirement for ATP in both these mechanisms of transferring material from the region of higher transport. So as we take a look at the two different types of transport, we'll begin by looking at that of passive transport. And as we look at passive transport, we said there's two types. We talked about filtration, so we're looking at diffusion. So diffusion, by definition, can be the net transport of material from a region of what? Higher to lower, so it's a net movement of particles, so it's a net movement of particles from a region of higher concentration to a region of lower concentration. The driving force for diffusion will be the kinetic energy associated with solid particles at temperatures above absolute zero. So the driving force for diffusion is the kinetic energy associated with solid particles at temperatures above or absolute zero. So it's the kinetic energy, the energy associated with movement, which drives diffusion. Particles, solid particles, that move from a region of higher concentration to a region of lower concentration. If we take a look to illustrate diffusion, if this was a beaker, this partition represents our artificial plasma membrane, and we have solution A on one side of the plasma membrane, solution B on the other side of the plasma membrane. We have a 70% solid concentration on A. We have a 30% solid concentration on B. We set the parameters that the plasma membrane is permeable to the solute. So based on the principles of diffusion, we know that the solid particles will move from what? A or from A to B? They will move from A to B down their concentration gradient. So our solid particles will move from A to B as they go down their concentration gradient, and diffusion will occur until what happens? Until we reach what? So diffusion, so we have the net movement of particles from A to B until we reach equilibrium. And what can we say about equilibrium? When equilibrium is reached, we have what? No concentration gradient. So at equilibrium, there's no concentration gradient. have equal solute concentrations on both sides of the plasma membrane. Are you okay with that? So we have no net movement of particles A to B or from B to A. We're not saying that we have no movement of particles across the plasma membrane, but at equilibrium, every time a molecule from A to B, conversely, another one will move from B to A. So we still have movement across the plasma membrane, but we have no net movement because we have them going back and forth at the same rate because there is a concentration gradient driving the movement across the plasma membrane. Are you okay with that? We want to point out, rarely do we achieve equilibrium for a particular solid across the plasma membrane of a cell. Rarely do we achieve equilibrium for a solid across the plasma membrane of a cell. And why is that? Then, okay, anybody, Elizabeth, you want to help them out on that? So we're saying rarely do we achieve equilibrium for a particular solid across the plasma membrane. Why is that beneficial to the cell? Not to have equilibrium for a particular solid across the plasma membrane. Because what are we doing? Diffusion is a type of what? Passive what? Transport. So diffusion is an efficient means of transport to get material what? Into and out of the cell without using what? Energy in the form of ATP. Are we okay with that? So rarely does a cell want to achieve equilibrium. This is not, there are caveats or exceptions to the rule, but rarely does a cell want to achieve equilibrium for a particular solid because it wants to have the capacity to drive that material either out of the cell without expending what? Energy. So we can use that gradient to transport something either into the cell or out of the cell. As we take a look at some factors influence the rate of diffusion, assuming that temperature remains relatively constant or within that homeostatic range. Take a look at some factors that influence diffusion. We can look at what's called Fick's law of diffusion, for Q, which is the rate of diffusion, be equal or proportional to that of our concentration gradient times permeability of the plasma membrane to the particular solid times that of the surface area of the cell. The rate of diffusion will be inversely proportional to molecular weight and that of the distance that the particle has to travel. So as we take a look at some factors that influence the rate of diffusion of a solid across the plasma membrane, assuming that temperature remains constant, we can see that the rate of diffusion is directly proportional to C, one variable. This refers to concentration gradient, and all we're saying is the greater the concentration gradient, the greater the rate of what? Diffusion. We said here we had a concentration gradient of 70-30. If we change it, we had 90% on one side and 10% on the other. We have a what? Steeper what? Gradient, and therefore these molecules will transport down the concentration gradient at a faster rate. So we're saying the greater the concentration gradient, the greater the rate of diffusion, ROD for rate of diffusion. So the greater the concentration gradient, or steeper the distance between solids across that plasma membrane, the greater rate of diffusion. Also, permeability. The more permeable the plasma membrane is to a particular solid, the faster that solid can what? Cross the plasma membrane and diffuse across the cell, either into or out of. We said the plasma membrane is what? Semi-permeable due to what? What makes the plasma membrane semi-permeable? The hydrophilic. So the permeability, the more permeable P for permeable, the plasma membrane is to a solute, the greater the rate of diffusion of that solute plasma membrane. Is this making sense? So we're saying if it's more permeable, we increase the rate of diffusion in which we transport material across the plasma membrane. A, the variable A represents that of surface area. So the greater the surface area of the cell, the greater the rate of diffusion. Surface area is determined by the what? Plasma membrane. So we're saying the greater the surface area, the greater the rate of diffusion. So the greater the surface area, the greater the rate of diffusion. This law is Alexander, right? How can cells increase their surface area? What do cells do to increase their surface area? Expand. What do you mean by that? Mr. Chamberlain. Increase volume. How do cells increase their surface area? What do they say? Yes, sir, Jermaine. Say it again. Villi. Microvilli. So cells are involved in diffusion or absorption or transfer material from one compartment to another. We would see a modification of the apical surface or the upper surface of the cell where they invaginate the plasma membrane with these microvilli. They increase the surface area. The surface area of the plasma membrane, and as we increase the surface area of the plasma membrane, we can increase the rate of what? Diffusion. Are we okay with that? So cells are involved in diffusion where we transfer material from one compartment to another. We see a modification of those cells on their apical surface where we have these microvilli increasing the surface area of the cell so to increase the rate of transport of material. Structurally, microvilli are formed by what? What is the structural component of a microvilli? They're made up of what? Microtubules. Actin, and actin makes up our what? Microfilaments. So make sure this is in that table. So they're made up of microfilaments, which molecularly consists of the protein actin. So we have the greater surface area, the greater rate of diffusion. Diffusion rate, or the rate of diffusion, is inversely proportional to molecular weight. So all we're saying, the larger the molecule, the slower the rate of diffusion, because we impede its interaction across the plasma membrane as it interacts with the hydrophobic core. So the larger the molecule, the slower the rate of diffusion. The variable x just refers for distance, and we're just saying the distance that the subsolute has to travel, the slower the rate of what? Diffusion. So when we think about diffusion, it's an efficient means of transporting material across the plasma membrane over what? Short distances. Are you okay with that? And it's very efficient because we're not using energy in the form of ATP. We're harnessing the energy associated with that of a concentration gradient. Now when we talk about net movement of solid particles from a region of higher concentration to a region of lower concentration, there's two types of diffusion. So we have two types. Of diffusion. We have what is called simple diffusion, and then we have facilitated diffusion. So we have two types of diffusion, simple diffusion and facilitated diffusion. When we talk about simple diffusion, Ms. Simmons, what do we mean when we say simple diffusion? Is it David, right? Yes. Okay. It's David, and right here, Danny? Danny? And David. So David, you don't know, Danny, you go ahead and answer. So David, what do we mean when we say simple diffusion? material that is what? Freely permeable to the what? Plasma membrane, so they can interact with the what? Hydrophobic core. Is that what you're going to say, Jermaine? So we're saying in simple diffusion, we're transporting material from a region of higher to lower, and the solid particle that's being transported is what? Freely permeable, P for permeable, to the plasma membrane. So therefore, in simple diffusion, we do not require a what? Transport protein. So it does not require a transport protein. So it does not require a transport protein. So in simple diffusion, we're transporting material which is freely permeable to the plasma membrane. It does not require a transport protein. An example of simple diffusion will be gas exchange between the lung-blood interface. So if you take a look at an example of simple diffusion, this represents a blood vessel. This will be the alveoli of the lungs, the functional unit of the lungs. We know that blood that's pumped from the right side of the heart, the right ventricle, to the lungs is said to be what? Deoxygenated blood. So this is deoxygenated blood returning to the lungs. It's deoxygenated. It has low oxygen concentration because our red blood cells' function is to deliver oxygen to the cells of the tissues of the body. And as cells undergo metabolism, they produce CO2 and release CO2, which is then transported by the red blood cells back to the lungs. So we have low O2 concentration, high CO2 concentration in the deoxygenated red blood cells. So this is a red blood cell. This is a blood vessel. In the lungs, we know that when we inhale, we're going to have what? High concentration of what? Oxygen, low concentration of what? CO2. So in the lungs, we have a high concentration of O2, low concentration of CO2. We said these gases are what? Freely permeable to the what? Plasma membrane. They're freely permeable to the plasma membrane. The capillary walls will be lined with a thin layer of simple squamous epithelial tissue forming the endothelium. The alveoli will also be lined with a single layer of simple squamous epithelial tissue. We said the single layer allows for the diffusion of the rapid transfer of material from one compartment to another. The CO2 will go down its concentration from higher to lower. Exhale, we release the CO2, which will be utilized in conjunction with water to produce carbohydrates by plants by way of photosynthesis. The O2 will go down its concentration from a region of what? Higher to lower. The O2 loading of the red blood cells. And we deliver this oxygen-rich blood to the cells of the tissue of the body. So we have simple diffusion occurring at the lung-blood interface, where we have a concentration gradient of CO2 and the oxygen. They're freely permeable to the plasma membrane. We diffuse down the concentration gradient as we have gas exchange occurring across the respective plasma membrane. Are you okay with that? Then we take a look at facilitated diffusion. When we say facilitate diffusion, what do we mean when we say facilitate diffusion? Okay, the concept, what do we mean when we say facilitate? What does that mean? Ms. Richardson. That's you, right? Renee, right? Yeah. Okay. Some of you are reading it, but we'll work with it. So take a look at facilitate diffusion. We know it's diffusion. We're going from what? Higher to what? Lower. So we're going from higher to lower. But in facilitate diffusion, despite the fact that we have a what? Concentration gradient, the substance is what? Impermeable to the plasma membrane. So despite the fact we have a concentration gradient which favors the movement across the plasma membrane, because the solid is impermeable to the plasma membrane, it requires a what? Transport protein. So the solid is impermeable to the plasma membrane, so therefore it requires a transport protein to what? Facilitate its what? Transport from a region of what? Higher to lower. So it requires a transport protein. So it requires a transfer protein. So we're going from higher to lower, but in this case we're transferring material which is not freely permeable to the plasma membrane. So we require a transport protein. We can look at two types. We have what's called channel mediated facilitated diffusion. So we have channel mediated facilitated diffusion, or we have carrier mediated facilitated diffusion. As we listed the other day, when we say channel mediated, we're just referring to a channel protein. Carrier mediated just refers to a carrier protein. These are two examples of transport proteins. So as we take a look at channel mediated facilitated diffusion, we have a channel protein which functions as a transport protein. So a channel protein is just a type of transport protein. We have our plasma membrane. A channel protein, if it spans the thickness of the plasma membrane, we call it a what? Transmembrane protein. So it's a transmembrane protein. Structurally, it will have a small hydrophilic pore which allows the passage of specific ions across the membrane. So this would be a small hydrophilic pore or channel that allows the passage of a specific ion across the plasma membrane. We know our ions are charged. They cannot interact with the hydrophobic core established by the fatty acids of the phospholipids. So we use channel-mediated facilitated diffusion to transport ions across the plasma membrane. We transport ions across the plasma membrane by using channel-mediated facilitated diffusion. If you take a look at the intracellular fluid and the extracellular fluid, the primary cation in the extracellular fluid is what? What's the primary positive charge in the extracellular fluid? Sodium. So the primary cation in the extracellular fluid is sodium. We have a concentration gradient for sodium. Is sodium higher inside the cell or outside the cell? Outside the cell, so we have a higher concentration of sodium outside the cell versus a lower concentration of sodium inside the cell, the primary positive charge inside the cell, or the primary cation in the intracellular fluid is that of what? Potassium. So the primary cation in the intracellular fluid is potassium, where we have a higher concentration of potassium inside the cell versus a lower concentration of potassium outside the cell. So if channel-mediated facilitate diffusion, Kelly, which way is sodium going? So in channel-mediated facilitate diffusion, which is a type of passive transport, which way is sodium going across the plasma membrane? Say it? Up the concentration gradient, like a down, right? Down. So in passive transport, channel-mediated facilitate diffusion, which direction is sodium going? So exactly. So in channel-mediated facilitate diffusion, because of the concentration gradient, sodium being higher outside the cell, being lower inside the cell, in passive transport, sodium will influx or enter the cell going down its concentration gradient. So in passive transport, sodium will influx into the cell. We take a look at potassium. At potassium, Michael, which way will potassium go? Yeah, well, that's all you said. Look at the concentration gradient. Based on concentration gradient, which way is potassium going to go? And channel-mediated facilitate diffusion, which is a type of passive transport. Just look at the concentration gradient. It's right here. So which way is it going to go? Which one? You only be in or out. Which one? Yes, sir. Will it go in? Look at the concentration gradient. Potassium's higher way. Inside versus what? Outside. So all together now, it's going to go? It's going to what? Efflux. So in passive transport, potassium being higher inside versus lower outside, potassium is going to efflux. It's going to leave the cell. So potassium is going to exit the cell, going down its concentration gradient. This is in passive transport. So you look at the movement of these two ions in passive transport. One is entering, one is what? Exiting. And it's the movement of these ions across the plasma membrane which will establish what's called a membrane potential, or a charge across the cell, which plays a key role in the functioning of nerve and muscle cells. So it's the movement of sodium potassium across the plasma membrane plays a major role in establishing the overall net charge across the plasma membrane. So we want to keep this in mind forever. Sodium is higher outside versus inside, potassium is higher inside versus outside, and in passive transport, sodium goes in, potassium goes out, and we'll see the opposite when we talk about that of active transport. We take a look at carrier-mediated facilitated diffusion, the second type of facilitation. So we have carrier-mediated facilitated diffusion, and in carrier-mediated, going from higher to lower, but we're transferring materials that are said to be what? Impermeable to the plasma membrane. The only difference, we're just using a carrier protein, also a type of transport protein. It's different in structure than the channel protein. It too is a transmembrane protein structure. It will have a small binding site, or it will have a binding domain. So it has a sequence of amino acids that bind to a particular substance. So it has a binding site that is specific for a particular solute, the binding site of the carrier protein increase affinity for that solute on the high concentration side. So we say, for example, this is extracellular fluid, this is intracellular fluid. We have our particular solute. We look at the mechanisms of action. Mechanism of action, MOA of the carrier protein. It will what? Bind to the solute. So the channel, the carrier protein, CP for carrier protein, will bind to the solute. And once again, we just want to highlight that the binding capacity for that carrier protein for its particular solute is enhanced when it's exposed to the high concentration side for that particular solute. Increases when it's exposed to the low concentration side. So the carrier protein binds to the solute. When it binds to the solute, what occurs? The binding of the solute to the carrier protein will induce a what? Conformational change. So the carrier protein undergoes a conformational change. And it goes from this orientation, where it flips, so to speak, from being exposed to the extracellular side, which is the high concentration side, to the intracellular side of the plasma membrane, which is the low concentration side. So we have a conformational change. It will then release the solute, so therefore transporting it from what? Higher concentration to that of lower concentration. So we release the solute. After the carrier protein releases the solute, what happens? The release of the solute causes a conformational change, or causes it to return to its original what? Conformation. So the carrier protein returns to its original conformation. Where it continues to what? Transport material across the plasma membrane from a region of what? Higher to lower. So all it's doing is just flip-flopping from one orientation to another as it binds a particular solute, transports that solute from a region of higher to a region of lower. An example of carrier-mediated facilitated diffusion will be a carrier protein called our GLUTs, GLUTs, for our glucose transporters. Glucose transporters are a type of carrier protein that transport glucose across the plasma membrane, either into cells or out of cells. We know cells need glucose to generate energy in the form of ATP. Glucose is a large polar molecule. It cannot what? Cross the what? Plasma